Now let's try to do this question that was asked in the second shift. Which of the following is correct structure of cytosine? Now there are basically 5 heterocyclic bases present in DNA and RNA. We name them as nitrogenous bases. Now these are basically purines and pyrimidines. The purines and pyrimidines, these are basically two types of heterocyclic bases. Now the pyrimidines are monocyclic, while the purines are bicyclic. The pyrimidine bases, they are monocyclic, while the purine bases, they are bicyclic. Now first understand how these purine bases and pyrimidine bases are really made up of. Purine bases, they contain a compound that is purine. Now let's first understand the structure of purine. Purine is basically a heterocyclic aromatic compound that consists 2 rings. So here in DNA and RNA we have purine bases adenine and guanine, and pyrimidine bases uracil, that is represented as U. Adenine and guanine, they were represented by A and G respectively. Next is thymine, that is T, and the next one is cytosine, that is C. So the purine bases are adenine and guanine, and pyrimidine bases are uracil, thymine and cytosine. Now how can we learn the purine bases? You can learn it by remembering AG, puri khani hai. So uracil, basically it is present in RNA only, and thymine, it is present in DNA only, while guanine, adenine and cytosine, they are found in DNA as well as RNA. So this can be sometimes also asked that which of the following is present in RNA. So our answer will be uracil. Which of the following is only present in DNA? Our answer will be thymine. Now let's understand this purine molecule. Now purine is basically made up of pyrimidine and amidazole. These 2 rings are basically fused together. So let's first understand the structure of pyrimidine and amidazole. Now pyrimidine is again an aromatic heterocyclic compound. This is the structure of pyrimidine. Now amidazole is a white or colorless solid that is basically soluble in water, and this is the structure of amidazole. So in the case of purine molecule, these two rings, pyrimidine and amidazole, they are fused together. So purine consists of two rings fused together. So let's draw the structure of purine. Now this is the structure of purine. If you carefully look at the options, and one thing more, that DNA and RNA they basically contains purine bases and pyrimidine bases. So the basic structure in the purine bases, that is adenine and guanine, will be of purine. So this is the common purine structure, and this molecule, and this is the common purine structure in the second option. So the first one is our adenine, as DNA and RNA they contain purine bases. So purine structure is the common in adenine and guanine. Adenine contains a extra NH2 group, while guanine has a extra double bond O. So A option is our adenine molecule, and B option is our guanine molecule. So these two are the purine bases present in DNA and RNA. I hope you have understand the basic structure of purine bases, adenine and guanine. Now let's move on to pyrimidine bases. Now pyrimidine bases, again we have already drawn the structure of a pyrimidine molecule. If you remember, we have already drawn the structure of pyrimidine. So basically pyrimidine bases are cytosine, thymine and uracil, and you can learn them by remembering CTU. So let's first draw the structure of thymine. Now as we have known that it is a pyrimidine base, so the basic structure will be of a pyrimidine molecule. There is a methyl group also. So this is the structure of thiamine. So pyrimidine bases, these are known as pyrimidine bases. The basic structure is of pyrimidine. So if we look at the C option, again there is a common structure pyrimidine present here. So this C molecule is known as uracil. There are two extra double bond O attached to a basic pyrimidine. So hopefully we have understood the basic structure. Again, if we look at the D structure, D option, there is a common pyrimidine structure in it also. So our C and D are pyrimidine bases and D is known as cytosine. It contains a NH2 group. Now this is very important point. Please remember all these 5 molecules, that is adenine, guanine, uracil, thymine, cytosine, they are aromatic in nature. Now in fact, one point to remember is also that the stimulants that we use in tea and coffee, that is caffeine, it is also basically methylated purine. And one thing more, there is a presence of guanine and cytosine base pair. And of course there are 3 bonds between guanine and cytosine. And there is a adenine and thymine base pair with 2 bonds. So this is again a very important question that may be asked, and there is hydrogen bonding present in these. So please remember that hydrogen bonding is there. So this is the whole detail of the heterocyclic molecules, the purines and the pyrimidines. And one thing more is that our caffeine is basically methylated purine. Now caffeine is basically a crystalline substance that is extracted from coffee or tea. So it can be extracted with the supercritical CO2. This is also an important question. So our question was, which of the following is correct structure of cytosine? So D is our correct answer.